Hey guys, welcome to topic 2 of chapter 20. In this topic we're going to expand from what we just talked about when we talked about forming of tissues to talk about how epithelial sheets are formed and the cell junctions that help cells communicate and hold together to form these epithelial sheets. So here's our topic outline. We're going to talk about types of epithelial sheets, then we're going to talk a lot about cell junctions. Here's our short objective list. Please let me know if you have any questions about these, and these are what I expect you to have memorized or understood by our next exam. Alright, so let's get started with types of epithelial sheets. So we talked about connective tissues a lot in the last topic. Epithelial sheets is a similar tissue that can be formed, but it's very different in what it's supposed to do. And so it is and you can see that we have a variety of different types of epithelial sheets that can form. We have the simple, we have stratified, we have columnar, we have cuboidal, and squamous. And this just depends on what we need these epithelial sheets to do. So each of these types of organization have their own function and their own types of communication between these cells. All these cells are held together through cell junctions, and these are in a variety of different ways, and each type of junction has its own purpose. And we're going to go through each of these here in the next little bit. But what you can see here in this image is that you see the cells, you see the junctions, and then you can see the basal lamina. And this helps hold those epithelial sheets in place so they don't go somewhere else or leave the group or even form a tumor by floating off and dividing. So let's start with tight junctions. Tight junctions essentially seal the cells together. So what it does is it brings them together really tight, and then it doesn't allow water-soluble molecules to pass between the cells. This is really important in our gut because what this does is it keeps all the nutrients and everything on one side of the cell so that we can use the concentration gradient to help push molecules across the cells through the transporters we talked about earlier this semester. The tight junctions are formed with proteins called claudins and alkylandins, and these proteins really help form that tight seal that you see there. And this also helps maintain cell polarity, as in which side it should be the lumen side, which side should be the basal lamina side. This keeps proteins from migrating all over the cell surface and keeps them in their very specific area that they need to be. Our next cell junction is adherens junctions, and this is one of the three junctions that's going to hold the epithelial sheets together. We're going to talk about the other two in just a second. This cell junction interacts with the cytoskeleton via actin, and you can see these adherent sections between the cells, and what that does is it holds them together, this bundle of actin fiber, and it uses these actin fibers to help really keep those structures together. The proteins that are used here are called cadherins, and they require calcium to help these bonds form. And actually cadherins are used in the other two types of structural junctions we're going to talk about. The big thing about adherent junctions is it allows the epithelial sheet to develop tension and change shape. So if we want like in embryonic development, things like that, that allows for the shape to be different than just a flat sheet. Desmosomes are another one of the three junctions that hold the epithelial sheets together, and you can see another image of it here. And once again, you can see the cadherins. Now each of these are using different cadherins. It's important to know that. This is just a family of proteins. But desmosomes connect to the cytoskeleton via intermediate filaments. And what this does is it creates the tensile strength you've seen in the epidermis, that nice firm strength that allows for a little bit of flexibility, but still some strength that allows for our outer skin. And so you can see how that forms here with this, with these keratin filaments that are intermediate filaments. Another important junction is the gap junction. In the gap junction, this is how cells can easily share molecules, either ions or water-soluble molecules in between cells. And what this can do is it can actually create a whole series of electrical signals or chemical signals that can easily pass between the cells that are connected through gap junctions. And you can see that here. They're essentially these little tunnels that will go between the cells and that allow for the cells to become connected. These tunnels are formed by proteins known as connexins. And you can see that here. And this is the only cell-to-cell junction out of all four that we're going to talk about today that it has a counterpart in the plant cell. Plant cells have a similar structure known as the plasmodesmata that allows for flow between the two cells easily. So this is also one of the few that allows for communication between cells, because we talked about tight junctions are blocking things from passing between cells. And so far all the other junctions we've talked about are all about how we hold the cells together. So desmosomes and adherence junctions help hold cells together. But what holds them to the basal lamina, which is important because, as I said, we want to make sure our cells the tissues stay attached to where they need to be. And these are done by hemidesmosomes. And these are such named because they look like half a desmosome. And you can see that here on this picture. And once again, it's really similar. It's connected to integrin molecules through the cadherins. And this is what holds the cells to the basal lamina. So this is the end of our short topic on epithelial sheets and the junctions that are used to help hold them together. As always, if you have any questions, please let me know.